Welcome back to Short Buzz. Today we are going to complete our bacterial genetics, which is your microbiology notes. We'll start with mutation. So I hope that you haven't heard mutation word first time because we are reading it since class 10 to 11th and 12th. So mutation is a sudden change in nucleotide sequence of the genes called mutation. Okay, nucleotide is our block of nucleic acids. Now mutation sometimes be very good, but often it can be destructive for the individual cell. Mutations are usually detrimental, but they can also lead to beneficial changes. Okay, as I explain you. A mutant is called an auxotroph if the mutation leads to new nutrient requirement, while the wild type strain is known as prototroph. So remember these two terms. Okay, if it's beneficial providing nutrient, it's auxotroph. Else, if it's wild, not good, then it's prototroph. So mutations are of two types, spontaneous and induced. In spontaneous mutation, what happens that it occurs naturally, about 1 in every million or 1 in every billion divisions. Like it doesn't happen to anybody. It's not that normal. And is probably due to low level of natural mutagens present in the environment. So mutation has to do a lot with the environment. With the change environmental factors, we get to see mutations. Coming towards induced mutation, it is caused by mutagens that cause a much higher rate of mutation. Like if a single person has get a lot of mutation, then we see this wild type. Okay, by chemicals or radiations. Other mutations are caused by some genetic element is transported. Now among these mutations there are also other paths, like it has divisions. We'll discuss further. So in types of mutation, the first mutation we will discuss is point mutation. Point mutation is a change in a single base pair of DNA by substitution, like it gets substituted, deleted. So deletion or insertion of a single nitrogenous base is what point mutation is. An example of point mutation is sickle cell anemia. It involves mutation in a single base pair in the beta globin chain of the hemoglobin pigment of the blood. Here I've shared a picture, you can see, but also do practice and learn so that if you forget, you can draw this chart. Point mutation is divided into three categories: a sense mutation, a missense mutation, and a nonsense mutation. So in our sense mutation, we get to see a substitution mutation, which results in the new codon, and that codon codes for both, like coding for the same amino acid. Whereas in our missense mutation, a single substitution mutation which results in one wrong codon and therefore one wrong amino acid. So here is the problem. Next comes our nonsense mutation. Here, a single substitution mutation when results in transcription of a stop or nonsense codon resulting in the termination of polypeptide chain. Next is frame shift mutation. So frame shift mutation is a gene that refers to the insertion or deletion of nucleotide bases in numbers that are not multiples of 3. This is important because a cell reads a gene code in group of 3 bases when making a protein. So these are mostly harmful because it leads to genetic disorder, which can be very bad in the particular person or any cell. So next topic is transformation. Transformation is a method of genetic recombination in which a naked DNA from a donor bacteria is transferred to a competent recipient bacteria and is incorporated into chromosome of the latter. Example here is Bacillus, Haemophilus, Neisseria, and Pneumococcus. A very short and simple, just learn it. So transformation occurs in nature. It is widely used in recombination DNA technology. In gram-positive bacteria, the DNA is taken up as a single-stranded molecule and the complementary strand is synthesized in that recipient. In gram-positive bacteria, double-stranded DNA is transformed. And there are several factors that affect its DNA size and state, competence of the recipient. Now, only DNA from closely related bacteria would be successfully transformed. Then competence factor and induced competence. I would suggest you to note down all the details for making it easier. So the last one is transduction. Definition is that gene transfer from a donor to a recipient bacteria. We're talking about bacteria. Okay, through a bacteriophage. It's known as transduction. Next is what happens that a virus infects here the bacteria. Types of transduction is generalized and specialized. Okay, hope that you understood the video. Thanks for watching. Do share, subscribe, hit the bell icon. Bye bye.